All right, welcome everybody to another one of our KBase webinars. Hopefully by now you've been on at least one of our webinars, but if not, we're trying to do these once a month to show a little bit more about what our system looks like and how you can use it to advance your science. So this month we've got Jose Faria from Argonne National Lab. He's going to talk to us a little bit about our metabolic modeling features and more so the advanced tools and science that you can do in KBase regarding metabolic modeling. Just a note, we are recording this webinar and we will post it on our website afterwards and send it around to all of you, so you can view it for future purposes, and feel free to share that as well. Hopefully all of you have a KBase account. If you have not made a KBase account yet, I would encourage you to do that. There's a link in the Q&A document. And so, yeah, as we're doing this webinar, encourage you to use the question and answer document to write down your questions that you have so we can respond to them in line. We've got a lot of people on the webinar today, so we don't really want to stop the flow for questions. So please just write them down in that document. I sent it to you all by email, but I'll drop it into the chat as well. And with that, I will kick it over to you, Jose, to lead us off.
All right. So I assume everyone can hear me. So very helpful. Thank you, Ben, for the introduction. So Ben just posted here on the chat. I think you may be able to see it because I'm here on Zoom, a link to the Q&A. Particularly with a lot of people here, it's going to be very difficult for us to stop and go and answer questions. So we have more people on the team, including people that develop these tools. I have not developed a lot of these tools myself. So other people can even give you better answers than I could. And if you post questions in the Q&A, even if we don't get to all the questions during the webinar, please refer to the document, because we should be able to, through at least the next couple of hours, answer all those questions. So feel free. So first on our advanced metabolic modeling webinar. So what we're trying to do here today is actually showing you some of our tools to come. I can talk a little bit what we consider more advanced tooling that we have, but we're actually excited to show you some of the tools we have in development. And even like it's an opportunity to get feedback if you were to be interested in trying any of our tools and prototypes that we'll show. I think this is it would be very excited to get you guys engaged at this stage of the process before we do major releases. So, and with monthly webinars, this is a great opportunity for that. So let me just talk a little bit about the basics. We're just going to go over the basics. We're going to look at a couple of workflows on microbiome annotation, modeling, and omics data representation in metabolic maps. And then very important is that share Google Docs. So I'm just going to talk a little bit about the basics. If you are familiar with KBase, you should be able to find, we have this tutorial that it's what we usually cover when we do a regular just kind of introduction to metabolic modeling in KBase functionality, where we show you how to build a metabolic model and how to run a flux balance analysis, how to import some phenotype data and do some of those simulations. So if you go to your dashboard and tutorial narratives and search for metabolic, you should be able to see that tutorial show up. So that's a great resource for you to get started. We were not going to cover a lot of those things that they're described there in detail. There's detailed text. I can open up to show you, but I'm not going to be covering that, but it's very useful material. So also upload your data. So this is also very important, is that we would want you to use your data. So if you want to do it, so in this case, we have 1 hour, so you may only just want to follow along. and not try to run things with your data. But this is something that is very important if you were to want to import your data, or if you already did. This is how we'd go about doing it. If we're getting data from our public repositories that are available, or bringing your own data to do the analysis. So this would be also useful. And then this is very important here today, that is this switch to beta. So we were aiming to do a release before today's workshop, but due to some technicalities, we wanted to make sure this was ready and really ready before we deployed it. So we have to switch to beta. So, and I show that again when I go to the narrative. So if you can see, if in the narrative interface, there's going to be like an R button you can press, and then you switch to beta mode, and that's where you can access all these apps that are in development. And it's just a way for you to even to see what it's upcoming in KBase, and maybe testing what other third-party developers are doing in the system if they have a beta release. So that's, I'm going to emphasize that too. And very important, you get the Google Doc QA, and please provide feedback when we say this is a prototype app. Please provide feedback because they'll be very important for the development of that app. So I'm just gonna step out here a little bit. So if you are familiar with the KBase dashboard, I don't know if yours looks like this, but I have kind of a lot of narratives going on. And the tutorial that I was talking about was this microbial modeling reconstruction tutorial, is like right here. And you can just like open it really quickly if you want, and you'll get something like this, where you'll be able to see what would be the how basically how to get started in KBase as far as building a model and doing some flexible balance analysis. Another cool place to go is if you want to go to the kbase.us website and go to research highlights. You can see some publications that some people have done using KBase, and they will be some cases of the most, I guess, advanced use of people have done in the system, and a lot of them are related to metabolic modeling. So you can, some of them would even have links to narratives where you can see the exact workflow that was used and try to replicate it if you want. So this is a good place for you to even look at other advanced workflows if you want. So here, when I look at that tutorial, you can see here that it has a little video even showing you how to build a metabolic model, and shows you kind of the capabilities that we have, and goes down as far as building a model and running flex balance analysis. So it's quite useful just to for you to have an overview. So I'm gonna go, so this is the document that we have. Yeah, everyone is, yes, we should, there is, everyone.
I can hear you, Jose. I'm not sure what the issue is, but
Okay.
check your Zoom sound settings, and check your computer settings, and make sure that they are selected to whatever you're trying to listen through.
I mean, can we have a show of hands? Just write in the chat if you can hear, just to make sure if it's only a few cases. I assume if you can hear me, just put in the chat that you can hear me. Okay. Yeah.
Most people
Good.
can hear. Thanks. Thanks, everyone.
Thanks, everyone. So one thing that I did here is, so there's this narrative here. That is what we're using. If you click here, this is what we're going to go over today, narrative links. If you click on that URL that we are right here, you should be brought to this. You should be brought to this Advanced Metabolic Modeling Webinar narrative. And what is going to happen, actually, it's going to look a little bit different from what it looks to me, because it's going to say that you can only see it in view mode. So this is actually, if you're not familiar with KBase, this is actually how things work. So since I made this narrative public to share with you, but as the author, I'm not giving you permission for you to change anything. So you can only view it, but you can't change anything. But if you want to change anything and kind of like follow along and tinker with the app parameters and even try to upload your data and replace what I've done, there should be like a handy button here, a copy button, where you can copy this exact narrative that I've done and rename it to whatever you want. And once you have your own copy, then you're free to go. Then you can do whatever you want as far as using this narrative in this data. You can play with the data, you can play with the apps, you can rerun the apps with different parameters. This is our big push for reproducibility. Ideally, someone publishes a narrative and you're able to go and if you really like their workflow, just replace it with your data and run it exactly like they did and make that effortless. So if you want to do that, here you'll have a copy. You click copy, you'll get a prompt saying, name your copy. And then it even tells you, do you want to open your copy? And then at the moment, you'll have full control of this workflow. So I'm going to get started a little bit. So first, just to get us a little bit of an introduction here, I would like to have like a show of if there's a kind of a show of hands in our chat, who here has attended a KBase intro webinar, basically a KBase where you were explained the narrative interface, how to use the narrative interface. So, okay. So I see I see a lot of no's. So I'm just gonna do a quick overview because, so we do this in a series and every now and then we do these intro ones. Okay, you have explored on your own, so that's useful. So basically, I'm seeing a lot of no's. So just a general sense of what's going on in here. So this is the KBase narrative interface. If you're just looking at my narrative, you won't be able to see. It just says view mode, and the data is kind of hidden like this, like the data panel. So, but basically, if you were to create your own narrative, just to figure out how things kind of work over here is here is your data panel. And so this is here, it's all where my data is that I used to do this workflow, and data can be imported by clicking on this. And in this case, you can see that I can import data from my own machine. You have example data here that is useful. You have public data. For example, we have like the entire VREF-seq. We have some other genomes. We have some public data here. Then there's the data that is shared with me. So basically, this is for example, people do one thing that you can see here that I can do is that I can share this with anyone if I go to sharing. So right now it's public, but I can make it private, or I can even share with anyone here. I know that my colleague Janica is on the line, so I can even share my narrative with him, and I can give him whatever permissions I want. So I trust him, so I'm going to give him permissions to edit and save and share. For example, and because of that, now I may have access to, instead of keep copying the same objects to the same narrative, I'm able to just, if I have someone shared something with me, as you can see here, I have a lot of narratives that have been shared with me. I can go anywhere and see, oh, there's a KBase, benchmarking. I was doing this benchmarking work here. So I can go there and try to get data from there directly without having to import it all the way from the beginning, just by adding it through the narrative. Or here, if that's a public, I see, oh, I really like this public genome here, I want to use it. Just by clicking add, I just added here, and now it's it's on my data. It's here, and then if I click in one of my data objects, it'll just pop up and show up here on my narrative, and I can browse this data object. In the case it's a genome, and this will be the same for any object. If you click it here, it will pop up in your notebook area over here. So this is very useful, where you can see here we have some text. So in this case, this is just markdown. If you double click, you can write some text. So the idea is that you can, instead of just running these applications, you would work to be able to write some notes. This would be our kind of your living paper, where you write some notes so that you won't forget. So 3 months from now, you come back to this analysis, you kind of don't forget what you did just by looking at this analysis at the apps. And then very important here is you have these apps here where you can click on this slide out, this arrow here, and it brings you a little bit of the catalog where you can look at the applications that are available. And if I scroll down, there should be a metabolic modeling section, and then you can see what it's available here. And just to use any app is just as simple as just clicking on it. If I click on it here, it'll just pops up in my narrative over here, and then I can fill up the parameters based on the data that it's available. So you won't be able to get the wrong data type in because we only allow the correct data types. So here you can only input genomes, you cannot put a non-genome file, for example, and so forward. So, and you can also search if you already know what apps you want. You can search here and see them categorized. And if you just one second, and if you even go to your KBase dashboard, which you should recognize as this here, and go to the catalog, you should have a better overview kind of on the larger page about what our apps we have available, in this case, metabolic modeling. And if you click on the eye, it should bring you to a page where it explains you all the documentation about it. So in this case, Build Metabolic Model, it's like a key app for us. So it brings you here. It's, for example, it has all the publications that were used and the relevant documentation. It even tells you how many times this was ran, for example, and it tells you the success rate. So it's over 60,000 times it was ran in the system successfully 93% of times, and the average run time. So just a little bit of an introduction here. So I'm just monitoring the chat a little bit. Yes. All right. So I'm now going to this was just a quick introduction. Hopefully this was useful for someone that never seen KBase before. And just trying to understand where we're clicking, where we're going. So basically, the other important thing here is to go over this, what I talked about in the beginning was this switch to beta. So if I click here, it's going to switch to beta. And there we go. And it tells you, warning, this exposes you to apps that are currently in beta. They may fail to run. So this is just so that you know that you're in beta mode, and then you'll be able to see apps that are not released yet. But for this exercise, since some of this advanced functionality, it requires the use of some apps that are in beta and that we really want to show you, and not just show you. We really want your feedback. Please use that Google Doc form as feedback so that you can help us with this. So in this case, what we did was we wanted to, this is a real problem that we see a lot of people trying to do in the system. We have a very comprehensive workflow right now for you to analyze metagenomes, from metagenome assembly and binning, and we have tools for QA and QC, such as Checkam, and to analyze taxonomic lineages in metagenomes. So I feel like we'll have a webinar on that very soon, I'm sure. But so that's something that we have right now, and people are bringing a lot of data on that, and then they want tools to analyze that data. So here, for example, is telling here that the data that we'll be using here came from this reference narrative. So you could click on it and go look at it. You can go look at how we analyze these metagenomes and did this binning. So and learn about that just by going there, by clicking here, it should take you to that. And basically what we did is I just used my data to go to this data share with me and my data to go and to that, to this narrative and just fetch the data that was relevant for me to do this workflow. So in this case, we're looking at a mixed isolate microbiome. This was a very interesting case to us at the time.
Jose,
Yes.
this might be a good, sorry, while you're mentioning this data, it might be a good chance to point out the published model org.
Mm-hmm.
I don't want to disrupt your flow, but if you show an orgs.
Yeah, I can show that. So also, yes, so we have done there is an in your organization here, you should be able to see on your dashboard, you should be able to see an orgs and organizations. So this is actually a new feature that we have where you can create organizations so that you can have your collaborators and narratives and works that you do. We see like there's lab, and you can make it public or you can make it private. We see that there are labs that are using this, like all orgs. So there should be here a published genome scale metabolic models org, and then if you click on it, you should be able to see what this is all about. In this case, you see these narratives, and here you should be able to see that there are some metabolic models that were already published that we imported into the system, and the analysis that we've done, and you can do analysis with those. So I think, thank you, Chris, for bringing this up, because
what I'd chime in and say is that there aren't a lot here yet, but we're adding more over time, and it can be a challenge to load published models in the system. And so this is a good resource to go to just check before you go and try and load a published model in the system yourself. You can check to see if it's been loaded here yet. And if you have loaded a published model in the system and gotten it to work, ask for permission to add your narrative to this organization, and we'll get it posted for you. Or if you built your own published model and you've loaded it into KBase, you want to share it, we'll load it in. So the idea is to have a single place in KBase where people can go to get the high quality published models that are available. But you can see here now, you have mouse and yeast and lactococcus, and I'll add the E. coli one from the Paulson lab to this pretty soon.
And as you can see, like, I'm actually not a member of this organization. Chris seems to be the only member. So I'm actually going to join it. So then hopefully he's going to approve me as a member of this organization, I hope. And once he does that, I should be able to have access to, not just have access to these narratives here, but I'll actually be able to request to add here to this organization and contribute. So if you'd like to be part of this organization, you can just go to organizations and find this one and then just request to join. So I'm gonna just jump back here really quickly. So we had this narrative where we did this, sequencing and analyzing and binning of these metagenomes. And here we realized this was a very curious case that we thought that this was an isolate. We thought that because it was replated like 3 times. And we just, and then when we did the sequencing, we realized there was just a community of 2 organisms, that it was just, it was a mixed isolate. So that we wouldn't know unless we were able just to, so we run our regular annotation tools, and we were noticing that things weren't working out. And then once we run a tool like CheckChem to analyze, we realized that we're actually dealing with a mixed isolate. So the first thing that we did is I went to this narrative that was shared with me, and I was able to find the relevant data. In this case, I needed an assembly. I needed an assembly to annotate with the RAS-TK. So what we've done here, if you're familiar with RAS to annotate genomes, we're using RAS to annotate metagenomes. So while you could be able to build a model, you want to build a model and then for multiple species, and then try to build different compartmentalized models and build a community model, which you can do in the system. Here, you can directly annotate a metagenome so that you can try to assess the overall, you may try to assess what are the capabilities of this metagenome, what functions can this metagenome perform? Is there any specific enzymes that are present, that are here, that are, should I be studying this community? So this, instead of you even before you having to bin and do all of these extra steps, you should be able to directly annotate a metagenome to start working. So, and this is what we did. So here we just, if you click here in view configure, you can only see what I've done. And here I'm just selecting an assembly, this assembly that you can see here. This is just a pure assembly object, just these metagenome context. And I just said that I wanted this to be annotated, just wrote it down, and then I get a result where I get this report where I can see the functions available, and I can even, you can click on view separate window to get a larger table, and you can search, and in this case you see there's over 20,000 features, which makes sense. It's around we know that it's 2 species that are in this metagenome. So that's how you go about, you'll be able to just straight away build annotate a metagenome. The reason we use REST, it's because if you're familiar with the modeling tools in KBase that use the model seed pipeline, we rely on REST annotations. So we rely on the REST control vocabulary to map reactions into functional roles. So that's why you have to annotate with REST. We actually have some very exciting work that soon we'll be able to share, where we, this was a suggestion and complaint that we heard from you, that you want to be able to use our tools to build models from metapsych annotations and KEGG, and a third-party developer has actually done this. They have successfully added tools so that you can do this, so that you can bring your own ontologies, KEGG or metapsych or even Go. You can even build models now from Go annotations and still use our pipeline just by mapping these ontologies. So that's a third-party dev that is working with us on doing that. So that's something that is like very exciting. So proc annotation can be, so you can annotate with proc annotation at this point cannot be used because proc has a different vocabulary. So we have proc in the system. Proc is available in KBase, but if you like, if I search here, you can see that proc is here. But to build models, you cannot, as of now, you cannot use proc annotations. We're working on that right now. We rely on REST annotations, or on in this case will be metapsych, Go, and KEGG that are tools that are being developed by a third-party developer. I know they're working on a publication as well to bring into the system to culminate that gap that we know that we have. So we annotated our metagenome, and then after we annotated our metagenome, we want to build a metagenome. We want to build this metagenome metabolic model. So very simple. We just find this. You just need to find the build metagenome app, which I did here. And like I mentioned, you just have to kind of click on it and just gets added to your system. And here, and then what I've done was you can just select you just have to select, in this case, my annotated. I annotated this metagenome. Here, reads for coverage. So basically what we're doing here is that if you have your reads, if you have the raw reads, which we do have for this, and you want coverage, so the idea here, if you're dealing with metagenomes, there will be some metagenomes that are more abundant than others. So this can be important information if you're looking at a metagenome model, trying to understand what species are more abundant or not. So by adding this information, you're able to capture this into your analysis, so that further when you're running model comparisons, for example, you're able to take this the coverage in account. So if you're trying to compare a model, see, oh, does this model have this functionality, this pathway active, and this one doesn't? And but how abundant is this particular species in my metagenome or not? That's why being able to add the reads for coverage, it's helpful here. You don't have to do it. It's not a required parameter, but it's something we suggest if you know what to do, if you have the reads and want to use it. So, and another thing that you said, see that we use here, we selected a media for gap filling. So in this case, we use carbon D-glucose. So all that I did was I clicked on my add data and I went to public. And then if I go to media, we have around, so we have like something like 500 media formulations here. So I just search for carbon D-glucose, which is a glucose minimal media, but you could search for others. So you could add it here to your system. You just add it to your, and you can look at it, for example, it's an overview. You can look at what compounds are present, and you can see the minimum and maximum uptakes. So in this case, and it's going to gap fill it with, is going to gap fill your metagenome model in glucose minimal media. If you don't want that, you can always uncheck the option when you're doing this. So I don't want to gap fill. I just want to draft. I don't want. I just want to see everything, and you don't do any gap filling. So that's also an option for you. So after you have your model, your metagenome model, you can inspect the results. You can look at what was added via gap filling. It will tell you here in this report. And then you can just look here and just completely export a model. It is telling you the reactions that you have and compounds and genes. So basically, in this case, we have kind of what we call a mixed bag model, because this is not compartmentalized. So this is not a model where you, for each, we're not replicating the pathways for both genomes. So you only have, if you go to compartments, we only have one extracellular and one cytosol, because we're modeling the metagenome as a whole, as what we called as a mixed bag. So this is a very different type. So that's why you only see like a smaller number of reactions total, because you're not gonna have any repeated reactions. You're not gonna have glycolysis shown both for each organism. It's just showing the overall capability of the metagenome as one community. But then what you can also do is, we actually know we can bin. We have this capability in KBase. We're able to actually extract genomes from metagenomes, which is what was done here. So we actually just want to build models for these isolates. This is what we want to do. We already know, we ran tools. We know that these are isolates. So we want to do that. So in this case, what I did was I just used this tool called the build multiple metabolic models. And I just went to, my other narrative where I had my genomes that are here, that are already nicely, it came from the previous narrative where they were analyzed and they were all annotated and whatnot. So you can get that. And here I did just same thing. I'm just gap filling them in carbon D-glucose. And you can see here there's the advanced parameters. We just went ahead and we gap filled. So the next step, which is very important, and this is like a new tool, is we call model characterization. So what is model characterization? So we wanted to be able to run basically a bunch of stats on a model to provide you some useful stats and information about the model. So all you have to do is just select the model that you want. And in this case, here, you can see here, we used one of these models that we just built here, this bin one of the bins that we built in the previous step. It's useful. Here you can see metagenome model with coverages. Again, this is not required, but if you have a metagenome model with coverages, it can become helpful to it can become helpful if you have a metagenome model with coverages for you to put that information so that can be part of your analysis, which species is more abundant in your metagenome. So in this case, we're showing you a toy case with two examples, but we know there's people working with like 50 metagenomes with like 50 species, and that gets really interesting, Like I can't wait to see some of those publications come out, because we have been working with some collaborators on doing some really cool analysis with these tools. Like we're developing these tools with them because they showed us that we needed these analysis. So we
do the read mapping when you build metagenome models, because we're working on also being able to use that coverage information in the flux balance analysis. So that's, you can't do it yet, but that's a feature that's coming. So it'll be useful to have that information in the model if you have it.
Right. So ideally, you'll be able to do some additional constraints in your flux balance analysis formulation based on the coverage for each like species in your metagenome. So that's one of the reasons too that we're doing that. And here you can see a little bit. You just get some statistics about reaction count and gene count, and you get some biomass. You get some ATP production, and you get some information about the essential reactions. And then you also get some pathway statistics here, and these will be pathways as defined by KEGG, where you get some information about the presence of reactions, if there were any reactions that were get filled in that pathway or not, and how complete or not the pathway may be. So these are all very useful statistics that you can get here. And then you also have this auxotrophy statistics, where I saw that recently just a paper from the Polson lab came out where they're proposing an algorithm for finding auxotrophy. And this is our kind of own take on that same problem, where we try to, we predict auxotrophy based on pathway completeness, and by being able to build models for a large set of organisms and using experimental data to inform our algorithm as far as pathway completeness, and try to tell you if your your organism is an autotroph for one of these compounds listed here. So when we release this, we'll have documentation with more details on the methodology, but this is what we're trying to accomplish here. So it's also all new features that hopefully you'll find useful. So we did this. So we characterize our two different models. And then we want to compare also. We have a metagenome model, and we have two models that we just made with from single organisms once we extracted these metagenomes. And now we want to compare these models. The problem with comparing anything, as far as depending on the metadata that you want to include, is that the metadata, it can get very convoluted depending on what we want to include. So we created this just little app that hopefully it's going to be more streamlined into the future. But we created this little app. I'm seeing something being drawn on the screen. Ben, is this just me or?
Yeah, someone, maybe you enabled it. I don't know.
I don't know how I'm doodling on the screen, but I don't know how, if I enabled it, I don't know how I did it. But because I wasn't even in that area when this doodling happening. So please, yeah, don't doodle on the screen. So we created this little app just so you can create a template for your comparison. So basically, what we've done here is we said from your current narrative, in this case, fetch a model object. So let's get our models from this current narrative, let's fetch them, and let's say what metadata fields are helpful for you. So in this case, we're interested in looking at completeness, at marker lineage, and coverage, because these are things that we can get from our metagenome analysis. But you can do whatever you want. So you could say, imagine you have data from the lab. You say like, oh, I know that one was treated in, like in a certain pH. And you know that some organisms in some of the models you're trying to compare they were grown at different temperatures, for example. So you can create whatever metadata fields you want here to use in your comparison. So you can fill whatever metadata, if this is free text, you can fill whatever you want. In this case, we went with these because these were useful for us. What we're trying to show with these was we cared about completeness, max lineage, and contamination. So, and once you run that, it creates an Excel spreadsheet for you that you can download. So if I click here, it's gonna prompt me to download an Excel spreadsheet that was gonna look basically like this. Let me show you. The Excel spreadsheet is gonna look like this. Just empty, just your model, and it just creates columns with the metadata fields that you gave it. That's all that it does. So, and then you can fill it with whatever metadata you want. So in this case, I have this marker lineage and completeness. So I have this, for example, I have this information for example, I already know that this was, for example, was incorrect in a sense because, okay, this is the completeness or this is the contamination. So this is my marker lineage. You see like how metadata can get messy if you don't know what you're doing. So, and here you can get your completeness, for example. And once I'm ready with my metadata, I import this file back into KBase. So what I did is I just use this little app here called import TSV or Excel file with these attributes. So that when I go run a comparison, I'm able to compare and look at statistics based on any metadata fields that I may want to compare. So once we successfully do that, we switch to doing this model comparison. So in here in the model comparison app, you can see that we're just using, all that we're using is this meta FBA model attributing mapping that we just uploaded. And when I ran it, I get this here that I'm going to click on view separate window because it's a very rich data set here. And so basically here you can see that I'm comparing the 3 models that I've done. I have my metagenome model and I have my bin1 and my bin2 models. And here you can see the pathway statistics. You can see there's functional reactions, total function coverage that you can see. And you can see at different models, you can see gap field reactions. You can look at gene counts. You can look at the average coverage per reaction, standard deviations. There's some very interesting statistics you can use. You still have your overall statistics here that we saw before, and you can search here. You also have some reaction classification statistics. But the cool thing here is that since you have that metadata label, for example, here we're looking at functional reactions, and you can choose this metadata. So like, oh, let me see how things work on the marker lineage or on the contamination or on the completeness. So whatever you have as your metadata here, you can filter, and you can filter so to make your comparison. In this case, we're looking at the z-score for the functional reactions in the model, and then you can also do the same thing. You can try to change the pathway label to a name or class. And at the end, after you have all this data, you can even download a CSV, or since this is a Plotly, you can even try to get your own image from here. So, and you can see here in this case, I'm looking at lineage. So this gets really interesting when you're looking at large metagenomes. So this is an app that would be great if you could provide feedback, because this is something that we can see improvement, like what are cool, useful statistics that you think you could show here. I think that would be great feedback on this app. But we're very excited about now we have an ability to have some visualization for you to compare your data, your models, and with your metadata to make it more meaningful and browsable. So you should be able to see any data point in this graph. So this is what we had here. So basically, this was just basically telling you how KBase is going about to serving the metagenome microbiome modeling community, what our strides we're trying to do. One thing that I didn't show you that you could have done is you could have just built a community model. Like, oh, I already know I have these two isolates. Can I just build a community model, a compartmentalized community model? And you can. Like, if you search for community model in metabolic model, you will have like merge metabolic models into a community. So I could have done that. So with my two bin models that I had in that case, which was this bin, my bin 1 and my bin 2, I could have just selected them. I could have just built a compartmentalized model by merging both. One cool thing that you see here is that you see this construct mixed bag model. So, but we don't want that because we already did that. Now we have this tool where you can go straight from the metagenome to build a model. So you don't have to do this. In this case, we'll build you a compartmentalized model with the which each organism is separate. And we also see a world now where we can bring these, the coverage, all of this information that we get from other apps to inform this community modeling tool to make it even more better for analyzing communities. So that's also in the making. So that's something you could do, that it's already existing and it's released. It's not a beta functionality. So, so this is what we wanted to show you on strides that KBasing is trying to make towards serving the microbiome modeling community. So other thing that we're looking at here now, and this is in prototype phase as well, and that we really like to show you is how to visualize omics data and metabolic maps. So in this case, we just went to this publication to get some metabolomic and transcriptomic data sets. That publication that is listed here. So we got the E. coli genome. We imported into KBase. The E. coli genome, once we were able to import the genome, we annotated with REST. Again, like I mentioned before, we rely on, as of now, we rely on REST annotations in order to build metabolic models into the system. So you have to annotate with REST. And after we're done building with REST, we just build a metabolic model. So this is a very traditional workflow that you can find in our tutorial that I showed earlier in the webinar. And once you build your model, you can then run flex balance analysis, something you're probably very familiar with. And here, I can show you a little bit as far as advanced. If I click on here, you can see show advanced, there's a lot of parameters that we're not using. So if I go here and do run flex balance analysis, so if I click here and just add it to my narrative, for example, I want to select a model. In this case was my gap filled model. You want to select the media you want to run. And then here, for example, if you want to change your objective function, in this case is maximizing biomass, but imagine you want to maximize the production of a compound. So you could try to, so you would select the transporter that is exporting that compound outside the system in order to optimize, for example, the production of that compound. So you could change that objective here. Then when you click on show advanced, you can see that flux variability analysis is being ran on by default on every time you run FBA. So minimization of fluxes. So we're doing here, we're doing PFBA. And then if you want to do all gene knockouts, that's a possibility here. Just by selecting this here. Here we have this where like you can click where we try to predict the a setting to predict the community composition based on when you're doing a community model. And then you could do gene knockouts. You can add multiple knockouts here just by selecting the genes, and you can remove. And then you would be able to do the reaction knockouts the same way if you rather knock out genes instead of reactions. And then you can add custom flux bounds. You can supplement your media. So basically, this is useful if you're trying to, if you have always trying to use the same media formulation, but then in different FBA runs, you want to try to supplement with different compounds. And then here, it's for expression. You can see here where if you have experimental data, you could add experimental data here, and you could try to set thresholds to set reactions as active as or inactive into to constrain your FBA. So this is similar to a lot of the algorithms then in the field that are they're very popular, where you use expression data to constrain flux balances, where we set thresholds and to call reactions on and off based on an expression data set. And you can also fix max carbon uptake, nitrogen uptake. So there's some advanced features that you have here. So please feel free to post any questions on those. So here we were not interested in doing any of that. We just did a very simple simulation on this glucose minimal media maximizing biomass. And then you can see here where you have all of your exchange fluxes, and you can see we have pathways, we have some CAG maps where you can see fluxes in the maps, but these are, we recognize these are not the most friendly visualizations, and we want more dynamic visualizations, and we want you to be able to analyze your data without having to parse through these complicated tables on exchange fluxes and reaction fluxes that, if you're not an expert, will be more difficult for you to parse through this information. And of course, we want you to be able to visualize your omics data too, if you have it. So in this case, we're talking about loading a transcriptomics data. So we had this data set from the paper, where in this case, you can even download it because we hosted it. And then what I did was that I just went here to import, and I just drag and dropped it here. And then in this case was E. coli expression. I said, I want to upload this expression matrix, and I click here to upload, and then it populates my narrative. It tells me, okay, this is your narrative. This is your expression matrix. If you already know, you may know what is the level transformation that happened to your data. In this case, we don't know, so you can leave it as unknown, and you name your expression matrix, and then you just hit run. So after you do that, you got your expression matrix into the system. You can even look at it. For example, here I can look at it, at my E. coli expression matrix that we got from the paper. And we'll do the same for, so you can look at the conditions. Here there were 6 conditions, and you can do like the features, and you can look at the expression here, data. And then we did the same for metabolomics data. So in this case, for metabolomics data, this is beta functionality as well, because only now we're interacting a lot with the metabolomics community. So you would have to use this in beta. Same way, if you search here, you search for chemical abundance, you should be able to find this import chemical abundance matrix imported into the system. And same way, you can download it from here. And then if I click here, you can see that file path. If I search for, in this case, if I search for matrix, I can see that I should have on my file, I have a, I have a cellular, I have this matrix that I can use now in import. E. coli matrix of metabolomics intracellular, and in this case, I know it's the raw counts, and I would import it this way. So this is a question I don't really want to get into, but people usually ask, like, why is it that you just hear it? Why is it that we're dragging and dropping in here and then using these apps to import things into KBase? Why can't I just put the file here directly? Why can't I just select here, pop up, and import the file from my computer? The reason we do this, it's because we want to capture the data as a data object so that we have validation, so that could be used across any other apps that use a metabolomics data set, and not just so that make that you need a specific file just for this app. So basically all metabolic, by doing uploading this way, adds you an extra step, but every expression matrix and every metabolomics data set should be used seamlessly across any apps that take those objects, instead of you having one single file type that you have to format specifically to run one algorithm, one given app. That's why you had this added step of having to run one app just to import the thing into the system, versus just getting your file directly here, just throw in the file path directly. That's why. So I recommend, if you're really interested in working in KBase and in working with these tools, to really look out for a webinar of Intro to KBase, because that's where Ben gives great workshops on that, where you'll go over how to bring your data and all these basics. A lot of these things here that may look alien to you, it really goes over those things that really make all of this seem easier once you understand how things in the system works. So, and then after you're done, you may want to visualize your data in maps, and together with your omics data. So in this case, this is, as you say here, prototype apps being developed. Please provide feedback if you play with these apps, or if you want to contribute to to how we're going about developing these apps. It will be very helpful. Testing, contributing with maps. So please do that. So here we're just looking at the TCA cycles, just a simple map here, where if I search for my Azure Pathway Viewer, I believe a lot of people here are familiar with Azure from the Paulson Lab, and we're progressively moving to use Azure as more and more in our visualizations. So you can find here your Azure pathway. So in this case, here you can see that it's telling you maximum column size, because in this case, we're only looking at one model, but you can do up to 10 side by side. So this is useful if you're trying to compare pathways and compare different models and pathways across different models. By putting more, you will be able to lay out multiple models side by side when visualizing. So that's really cool. And then if you're doing gene expression, you can select it here. And then when you click here in grid cells, we'll ask you what map catalog you want to use. So we have some maps here that we've been working on, and then we'll be working in adding more. And then you can build your own map and import it into KBase if you're familiar with Azure, which is something that I'm going to show you too. But I can just select here the model. I'm looking at this E. coli model. I have FBA that I want to visualize on my map. I have a chemical abundant matrix, and I list the condition would be, I think there was a glucose FKM, and I'm not sure, but it's filled down below. And the same thing with, I have an expression matrix. My favorite expression condition would be the one I want to use, for example. And then, so if you look here, and it's already done. So basically, after you list all these parameters, you get as an output, you'll be able to get your map. And in this case, we're looking at this TCA map. I can even bring it over into a larger window. So basically, so here you can see that this is in our IDs, but you can switch here IDs to names. You can save from here. If you want to save this map, you can save your map into if familiar with Azure controls, you can still use Azure controls here. You can save to map into your machine. There's a lot you can do here, but more important is, so basically here right now is just showing you the flux. It's showing you the flux through the TCA cycle. Darker color means that you have more darker. It means that you have a higher flux through that reaction. So we're still kind of working to tune this up. And then one thing that you're already seeing here is if I do expression, you can see here now, instead of flux, I'm showing the gene expression, where there's more reactions with that show the higher expression here. So you can switch that overlay. And another thing that you can see here too, you see that some metabolites are highlighted. So basically this is the result of the metabolomics data set. So what is happening here is telling you that in this case, there's succinate, which makes there's higher production of succinate. And here, I guess, as a cofactor, phosphate was like the compound that was more chemical abundant in this data set. So you can see here, there's different degrees of size of the metabolite and color that will indicate the chemical abundance, but in this case, it makes sense that what we're producing as a result of this is succinate, so we have this here more highlighted. So this is still, as I mentioned, still in prototype, so any feedback that you can provide here will be useful. And if you were to want to, as we are at the 1 p.m. at the hour mark, if you were to want to import a map, design maps yourself and import a map as a JSON file, that is the format for Azure. We also made this importer, you just import your JSON file. And same way, you do exactly as you've done before in order to get your pathway. In this case, because we wanted, there was no aromatic amino synthesis map, there was just individual amino acid pathway maps. So what we did is we're able to import the map into our narrative, and then the only difference is when you look here, we just say that in our map catalog, oh, we're using a narrative workspace map. We're using one that we got here, and then you just select, oh, okay, this is the one that I imported, that it's here, and then it's exactly as it was before. And when we look at the result, when you look at the result here, you have the same as before, and I can go to full screen. For aromatic amino acid biosynthesis, same way, you have, I can switch to names, and here you would have the flux by default, or the expression data. We're going to definitely want to tweak these colors and overlays, and also you can see the chemical abundance on what metabolites show more abundance in as far as being produced. So this is what I had. So I Ben, I think we can. I don't know what's the procedure. Do we stay on for questions? But this is what I just like to thank you everyone for being here. I believe this was the highest attended webinar we ever had.
Yep.
Right. We kept.
Yeah, so if
we kept Zoom. I think we have next time we have to pay for Zoom. We have to pay them more money.
I don't think so. I think we actually can do up to 200. But
okay, good.
Yeah, if there's any open questions or unanswered questions on the Q&A doc you want to go over, that's totally fine. Otherwise, we can wrap up.
Okay, let me just go over here a little bit, but basically, this is what we had. We wanted to show you a little bit some of the things that KBase can already do, and more importantly, engage with you as a community, like where are we going? What are the tools that are coming? What is it that we're developing? And more important, did you see a tool that you're developing that you think like, oh, there was a step missing right there? There was an analysis here that it could have been done, that I have a tool that would be great to even further improve this. Like particularly if you are a developer in the system, I hope that you were familiar with the KBase SDK. If you search for KBase SDK, you can be brought here for our KBase documentation, when there's extensive documentation on how to bring your tool to KBase. And we'll give you training. You can join our developer Slack, so you can get help directly from our devs. We can even give you. We can be on a call with you. We can provide training if you have a couple of people in the lab that want to be trained on the KBase SDK. So it's a great way. We have success. As of now, we're having more and more apps that we did not develop. There's third-party devs that realize that they have a functionality that we did not implement that they want to see in the system, and they come to the system. So I highly incentivize people to do that, and engage with us, because we can help you. We can help you bring your tools to KBase as a community, so that we can make the system better for everyone. So, is there any questions here that are unanswered, or I see that guys are working on it? No, I think everything is answered, correct? Okay, I see people interested in Cobra Pi. Really like the workflow, but I'm very used to love these modeling techniques in Cobra Pi. Cobra Pi scripts, we want to progressively move more closer to CobraPi. We did not show that. For example, one thing that I can, if you're familiar with these tools, as far as this being an advanced metabolic modeling webinar, for example, when I build a metabolic model, I think this is something that should be a part of a advanced metabolic modeling webinar, and you want to build multiple models and you don't want to, and want to tweak the parameters. For example, if you look here, if I click here on this show code over here, if I click here in show code, you can see that basically you have all the parameters, basically our API here on how do you go about running this in KBase. So in this case, if I were to copy this piece of code here, for example, and if you see here, there's a code. So this is KBase is built on top of Jupyter notebooks. So you can still run code here. So I would be able to say, for example, this is a build metabolic model. I just want to change the output, new output, new, and change the, you can change the parameters here and you can iterate. And then if I just do and if I do shift enter, I would just be able to immediately, you can see that I'll be able to run this piece of code. And once I'm able to run this piece of code, oh, it's telling me that I have a syntax error. Oh, of course, because I forgot I'm not closing my parentheses. So if I go here, so now once this runs, you're able to, you can see now that your code cell, it was converted into a job. So it's in the queue and it's going to run, and then you're going to get an output from this. So you can even create complete workflows where you fetch from all multiple apps, and you can kind of cascade run multiple apps that may require. If you're trying to do that and is not succeeding, please let us know so that we can help you with that. But that's if you're familiar with the program, if you're programming skills and scripting, you can do that. So there is a world where we could progress towards having CobraPi more integrated into the system, and you'll be able to run similar CobraPi scripts in KBase as well and take advantage of all the other functionality in KBase. So this is something that you can do here right now. You can write a piece of code that annotates a genome, builds a model, runs FBA, and runs model characterization, and gives you, and you even can write some code to parse all of that into a table that you can analyze and get a little bit of data from every output object to produce exactly the output that you want. So that's something that you can do as far as more advanced functionality. So so here, can you explain how to read the exchange compounds in the table? Question, why have you included a model seed in the reason why have you included a model seed in your toolbox but not CarveMe? So we could, we have the model seed because it's basically because we are part of the team that developed the model seed. So that was the first one. And like any other tool in KBase, there's really no preference in most tools. Is we really can't import every tool. Like, so I give a lot of webinars, a lot of conferences. I go to a lot of conferences, give a lot of workshops, and everyone always wants to come and say, I really like KBase, but that this algorithm that I really like that you don't have, can you implement it? And the answer is, maybe we can. If there's a lot of demand, if you go to our KBase help board, which is something that I'm also going to highlight right now, you can go to the KBase.us and you can go to the help board and make requests and for features. And if a lot of people subscribe to your request, a lot of people comment and say, I really think this is a valuable feature, maybe that would happen, but it's mainly resources. So we have one tool. You say CarveMe, I would say there's multiple toolboxes that we could have, multiple model reconstruction pipelines that we can have but we can't have them all. We love to work with the CarveMe developer to bring CarveMe to KBase, for example. That would be great.
Third party dev is supported. We have an SDK. Anybody who wants to can add an app to the platform. So the other thing I'd suggest is if you want a particular app like CarveMe, you can also ask in KBase. You can also ask the CarveMe devs about this, and hopefully that might encourage them to reach out to us and work with us. Because if we have to wrap everything ourselves, that takes a lot longer than if the devs really work with us to get those tools in.
Right. And for example, I know that some tools in this field, for example, we have a challenge where if they require the use of, for example, Cplex, we cannot use Cplex because Cplex is not open source. Cplex is a tool for academia, only for academia. And since KBase is an open platform, we cannot guarantee that a commercial company is going to come into the system and use the tools. So we cannot use Cplex or Gurobi into the system. So we're looking at, we're able to use Skip at the moment, but that's a limitation that we have. I know that in the field, a lot of times, since those are free, everyone just defaults to those, and they should, because it's Cplex, from all my experience, it's always yields the best results. But we can't use it. So if there's a tool that heavily relies on that or is developed specifically for that, then we have to, again, talk with the community. I know that we've been talking, for example, with the Optlang guys as far as trying to get other alternatives for MILP solvers that are not Cplex or Gurobi, so that we'll be able to get more tools in that space in the system. So that's one challenge that we have. So is proteomics, metaproteomics data supported for kinetic metabolic models? Not yet. So not yet, but this is something that it's in the pipeline. We're definitely developing, we are working on this, mainly because these are capabilities that we really didn't have in our group as far as kinetic modeling. So we now have someone working on that. But this is another field where we think that the community could really come in and help, because it's about a lack of expertise, in a sense, from our dev side, of not being experienced with these tools. But we love to work with you and learn from you and how to bring these tools into the system. So that will be great. We are working on something ourselves, but to really get this going in KBase, this is something that definitely would require a lot of help from the community.
I think we're also hoping to add ETFL to this, to do some of these proteome-driven dynamic models.
Yeah. So I know that there were some of these latest modelings that we've been seeing, that we are working with some devs to bring their proteome, proteome and expression, protein allocation and expression models into the system. So everything in this sense, like here, that you see that is possible, but it's about working with the community. Because if you go into the system, if you go into KBase and look at our catalog that I was showing you earlier, it's just impossible to do everything. So we're not just focusing on modeling. If you look through our catalog, we do reads processing, genome assembly, genome annotation, comparative genomics. So there's a lot of pieces in the system that we have our team touching and developing. So to really, we really need the community in any of these fields in order to help us push further. So we would say, as far as algorithms, we could get one or two ourselves to get the analysis kickstarted. But if you really want to see your favorite algorithms, you'd have to hopefully work with us to come into the system so that we can develop. For example, we know that, for example, a memote was just published, and a lot of people here were involved in that, and we worked with them from the beginning to be able to have memote in KBase. It's not ready from prime time yet. We're working with them, but we already have a beta version, for example, where you can select a model in a media to get a memote report here in the system. So this is an example of us engaging with the developers, saying, like, we think your tool is important. We want it into the system, so let's work together. So they did work. They did some work. We did some work. So we're able to get this done. So this is a model that we feel like it's how we're going to advance our metabolic modeling capabilities into the system, will be through this model of joint development. So let me look at the chat. Can you generate metabolic map using metagenome and metatranscriptome data? You should be able to do this, yes. Right, Chris? As far as using
there's you can visualize a metagenome model, and I know we described the workflow both in this Q&A sheet, and Jose demoed it as well.
Yeah.
You can build a metagenome based model from a metagenome assembly, and then once you have that, then you can use the pathway app that Jose showed to viz that model. It doesn't yet show coverages painted on the map, but that's something we're thinking of adding. But yes, you can also add metatranscriptome data to that model. Okay, yeah, visualizing metatranscriptome data on a metagenome model is going to be a problem.
Yeah.
Now that I think about it, the problem is the metagenome models do not contain the gene IDs associated, because I guess once we've enabled the ability to add coverages to the maps, then you'll be able to do it. So you can't do it yet. The problem is we don't keep the gene IDs in the metagenome models, because that would make the models too big, because you have metagenomes that can routinely have 1 million genes in them. And a lot of those are going to be metabolic. So the models just explode in size and it breaks a whole bunch of things. So even if you had a feature value matrix for a metagenome, which you can't make with the RNA-seq pipeline in KBase because it only works on single genomes, you still wouldn't be able to paint it on a metagenome model map.
Right.
You can paint transcriptome data on, you can align metatranscriptome data to a MAG, to a metagenome assembled genome, a bend metagenome, and then paint that transcriptome on a map. Or like I said, we're working on adding coverage painting on a map. So you could, when you, once we have that, you'll be able to map your reads to your metagenome assembly as coverages and load that into the model, and then you'll be able to paint the coverage of the metatranscriptome to your metagenome.
Right. So I'm seeing someone here said they may have more questions later. Yes, you most definitely can. So at the Q&A doc at the top, there's my email. So feel free to email me with questions. Or you can use the KBase help board as well. So as I'm going through here, but yeah, so like Chris touching a really good point. Just you came to the metabolic modeling workshop, and maybe you learned something about other tools that you could be using in KBase, for example. Yeah, something that we didn't show here. So when we look at this advanced, so here we uploaded an expression matrix directly from a paper. But if you had your own expression data, you could have done everything in KBase. You could have like started. If you look at expression analysis, we have a full suite of tools. So you could have brought your reads, you could have done alignment, you could have done your expression data analysis to then run with your model. So a lot of these steps that here we went and got from that paper, you could have done a lot of these steps already in the system as well. So branch out to that as well. So one cool thing that I like to show usually is, this is kind of an intro to KBase thing. Like on any if you click on here, you should be able to see, if I go here and do, for example, you can click, for example, here saying this is a model, and you can see show apps that with this as an output. So you can see it will filter here the catalog with all apps that output this. Or in a sense, like show apps that use this as an input. So you have a model, you want to see what is it that I can use in the system. By clicking here, it filters basically every single app that will take a metabolic model, so you can see what it's available. And this is for every, in this case, like expression matrix. If I show apps with this as output, by doing this, I look as expression, and you can see that there are expression tools that will create this for you in KBase. Or if you look at it as an import, you can see that you can do a lot of other stuff with your expression data as well. So it's a useful thing if you're just trying to figure out what the system does and you find the catalog a little bit overwhelming, because the tools are growing, and particularly because we're getting more and more third-party developers in. So I feel like we've been focusing a lot on making the platform work and be more reliable. You can see that we now, like, you can look at your jobs and your improve statistics. You can look at we're improving our queuing. We have feeds for you to can be aware of if what's going on in your organizations and your narratives and what your collaborators are up to. So we're improving our search. We created orgs like we showed. So we're doing a lot to improve the reliability of the system and to make it and hopefully improve the user experience. And the community is helping us bring new functionality. So I think we should wrap up now, unless there's anything else that is missing. Because I still see 40 people here. So thank you for staying, guys. I'm surprised still so people have still stayed to listen to me for an extra 20 minutes. All right. Is any question that we're missing, Ben, you think? Ben?
Hey, I'm signing off.
Okay, signing off. All right. So thank you, everyone. And we'll still go through if you have more questions, please go here.
I'm stopping that other webinar right now.
No problem.
Okay.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE KBase. And if you have questions or encounter an error when using KBase,